in amyotrophic lateral sclerosis. He has served as an elected trustee of the World Federation of Neurology. Currently is the first vice president, and he's also served on the International Executive Committee of the Movement Disorders Society. Dr. Kaji is also an executive member of the Japanese Society of Neurology and the Japanese Society of Clinical Neurophysiology, and a member of the International Federation of Clinical Neurophysiology. You're welcome. Dr. Kaji, you present amyotrophic lateral sclerosis, diagnosis and management.
Thank you very much, Professor Nisserine. May I start sharing my screen? Yes, I'm Ryuji Kaji from Japan, and I'm basically an EMG, a clinical neurophysiologist, and I like to talk about early diagnosis of ALS and the development in the field of management of ALS. And later on, you will listen to Professor Jacobs. He will just talk about probably a very new method of treating ALS. Now, let me start my part. And this is my conflict of interest disclosure. And as you all know, that amyotrophic lateral sclerosis, this is English, sorry, not French, but this was first described by Jean-Martin Charcot. He's a French founder of neurology, and more than 150 years ago, and almost 150. And he has described a patient who has muscular atrophy and at the same time having a pallor of the lateral column of the spinal cord. That means this is a part occupied by upper motor neurons, and spinal cord anterior horns are also depleted with neurons. And then this means lower motor neurons. So upper and lower motor neurons are gone at the same time compared to the normal spinal cord. So this is the reason why he coined the term amyotrophic lateral sclerosis, ALS. And the clinical features are progressive loss of upper and lower motor neurons, as I have said, and lack of sensory involvement. Usually, some rare genetic form have sensory involvement to a minor degree. And very peculiar finding is ocular movements abnormalities are lacking. So even though there's no muscle in the body, eye movements are spared at the very terminal phase of ALS. Of course, there are some exceptions. And it is also interesting to note that lack of sphincter abnormality or dysuria or just the bowel movement control of the sphincters lacking. The disturbance is lacking. So that means if you have spinal cord compression, for example, for cervical spondylosis or any other traumatic cause of spinal cord injury, then you have at first sensory involvement in at least in the lower limb, and at the same time, most of them do have urinary urgency or dysuria. But lack of sensory involvement and lack of sphincter abnormalities tell you this is something different from spinal cord injury. And also, this disease presents with fasciculations, twitching of the muscles. So this is also characteristic. And this is a video of fasciculation, the very terminal phase of the disease. You see prominent fasciculations all over the muscles that you can see, which are atrophied. And so there are many genes causing ALS. And our group have found optineurin here as one of the causes, but there are a number of different mechanisms probably, because all these genetic abnormalities are behind. And ALS is not probably a uniform disease, but common final path with heterogeneous causes. This is my view, but Professor Jacobs would mention more details probably. And this disease is also called as Lou Gehrig's disease. And Lou Gehrig is a very popular baseball player. So I think baseball is not very much popular in French countries, Francophone countries, but in Japan, baseball is very, very popular. And recently, a Japanese baseball player went over to the United States Major League, and you might know Otani-san is now one of the many favorite baseball players in the major leagues. And Lou Gehrig developed finally ALS, and that was the cause of his death. And he died at the age of 38, as you can see here. But he was just playing baseball. You can just see atrophied first dorsal interosseous muscle here. And at this time, he still was one of the just having a large number of home runs. And so disease process begins well before the serious outcome of the ALS occurs. And then the disease process starts very early in the course, even though the obvious clinical onset. Recently, Alzheimer's disease is now being tried and even developed as a clinical disease-modifying therapy. And some of these Alzheimer's trials employ a very early detection of the disease using PET. So even though those Alzheimer's patients do not have full-blown disease process, the disease-modifying therapy should start very early in the course, even before the obvious clinical onset. The same probably applies to ALS. That's the main message of my talk. This is a very old study from Japan, published in Neurology a long time ago. And this is MUNI, motor unit number estimate of the patients. And this is the control and ALS, and this is Appel score. So these are just increased. And this is a function of the patients. And this is MUNI of ADM. AB-ductal digiti minimi, and this is MUNI of extensor digitorum brevis. And these numbers are significantly decreased even within the first year of the clinical onset. So after 1 year, just 1 year, those patients lose more than 70% of the motor units already. So if you start disease-modifying therapy at this moment, it might be too late. So that's the message. And so there has been diagnostic criteria. The most famous one is Escorial criteria, and endorsed by WFN, where I'm working right now. And this is a very good criteria, very famous criteria, but this is only for research purpose. And so these research criteria are used for clinical trials. And if definite category is fulfilled, more than 50% of lower motor neurons might be lost. So there have been many, more than 100 clinical trials failed in the disease-modifying therapy for ALS, but many of them are too late for any therapeutic intervention. So I pondered that there has to be a good way of detecting this disease very early in the course of the disease. And at first, let's review Escorial criteria. We divide the body part into 4: bulbar, cervical, thoracic, and lumbosacral. And then in each regions, we define upper and lower motor neuron signs. And which is very important is by clinical and laboratory assisted, that is mostly EMG. And the disease process has to be progressive. And what is more important here is excluding other explanations. That is basically, this is the exclusion or excluding other conditions. And the diagnostic certainty is definite, probable, possible, or suspected. And the suspected only lower motor neuron signs in 2 or more regions. So this might be mimicked by various other causes. And possible upper and lower motor neuron signs in only 1 region, or upper motor neuron signs alone in 2 or more regions. Lower motor neuron signs rostral. Rostral means if you have lower motor neuron signs in the upper arm, then upper motor neuron signs in the lower limb. But this is also mimicked by cervical spinal cord injury, as I have mentioned. And diagnostic certainty is further subdivided into clinically probable, and this is upper motor neuron and lower motor neuron signs present, but upper motor neuron signs rostral the other way around to lower motor neuron signs. Muscle atrophy in the lower limb and upper motor neuron signs in the upper limb. So you cannot explain these signs on the basis of spinal cord injury or some other disease. And clinically definite, upper motor neuron signs, lower motor neuron signs in 3 regions out of 4. This is rather complicated, but this is very rigid. Rigid, but probably too late. And then just being conscious about the more use of sensitive method, Arihaus revised criteria to Escorial was published in 1998. And so clinically probable laboratory supported ALS, this new category was defined. And lower motor neuron signs defined by EMG criteria and fibrillation and positive sharp waves. This is a great jump in the definition of the disease. And this is means active loss of lower motor neuron innervation to the muscle, and at least 2 limbs, and proper application of neuroimaging clinical laboratory protocols, so on and so. And no need for upper motor neuron signs rostral to lower motor neuron signs. So this category is a rather over expansion of the previous classical Escorial to some degree. But most of the diagnosis is in the hands of EMG at this moment. This is fibrillation positive sharp waves. This is just at rest and use you hear the pseudo-random buzz of the muscle fibers lacking motor nerve innervation. And then at the end of the executive period of my IFCN period, we organized a meeting, expert meeting in Awaji Island, which is next to Tokushima, where I serve as the chair of the neurology. And Awaji, then it was called Awaji criteria. And this is a very special diagnostic criteria for early diagnosis. And this is just designed for the use of clinical trial, just employing more patients at the early stage of the disease. And this is in Awaji Island, 2006. And then we put together all opinions, and then we defined ALS electrophysiological way, and we put fasciculation potentials equivalent to fibrillations and positive sharp waves, and this is a big jump in the electrophysiology. And we also used unstable and complex motor unit potential as one of the key features of the electrophysiological findings. And unstable motor unit is defined as just a very large jitter if you do a single fiber EMG. This means neuromuscular junction is becoming in failure. So this reflects the ongoing degenerative process of the motor nerve endings in the muscle. So this is also equivalent to fibs and positives, as you have seen. And also fasciculations. Fasciculations are very irregular and usually complex and very bizarre looking. In ALS, the frequency of appearance is rather low. So we put Awaji criteria with early house, and then with these new electrophysiological features, we strengthened early house criteria for Escorial. And so in clinically probable ALS and clinically possible ALS, and clinically probable, as I have mentioned in early house revisions, upper motor neuron signs not necessarily rostral or above to the lower motor neuron signs. So this is just early house interpretation of Escorial. And lower motor neuron signs rostral to upper motor neuron signs. We also just erase these parts. And these Awaji diagnostic criteria became unexpectedly very popular in the literature. And this is from Annals of Neurology. And there are many studies later on. In short, Awaji criteria made it possible to diagnose more than 6 months earlier than Escorial or early house, and approved by meta-analysis. So more recently, we got together in Australia and Gold Coast, and this was organized. I'm here. This is organized mainly by Australian people, Matthew Kiernan and David Burke. And so this was published in Clinical Neurophysiology as well. And this is the essence of this new criteria. And so this is a matter of excluding other disease processes, so exclusion diagnosis. And the presence of upper and lower motor neural dysfunctions at least one body, and with upper and lower motor neuron dysfunction or lower motor neuron dysfunction in at least two bodies. So this is very flexible diagnostic criteria. So most of the process of diagnosis is exclusion of other diseases. So this is much more simplified, and I hope these criteria will become more popular. And so, so most of the diagnosis is at the hands of BMG as now. So what is most important in the electrodiagnosis is any signs of demyelination should be ruled out. I'll tell you why. The variant syndromes, you have to exclude various others. For example, progressive muscular atrophy, which some of them do have curative disease therapy, and primary lateral sclerosis or HSP, and some other diseases. Also, Kennedy's disease, bulbospinal muscular atrophy. And what I'd like to mention is demyelinative conditions that mimics ALS is multifocal motor neuropathy. This is characterized by conduction block, and half of these cases do have elevation of anti-GM and IgM antibodies, which is kind of autoimmune. And this is one of those patients. He originally had a diagnosis of ALS. As you can see, fasciculations over the shoulder muscles, pectoralis major. And it was very interesting to see if you do manual muscle testing in detail, flexor digitorum profundus muscle, digit 2 and digit 3 are not very weak, but the same muscle of digit 4 and digit 5 rather weak. So there are quite a distinctive difference of the muscle power between these two fingers and the other two fingers. So this means this muscle is innervated by median nerve for these two and ulnar nerve for these two. So this is a smell of peripheral nerve involvement as well. So that's the key to the diagnosis. And then this is the original nerve conduction study a long time ago, 89, which I discovered IVIG for the first time to treat this. And this is after stimulating at the wrist, and this is at the elbow, and this is at the axilla, and this is Arab's point at the shoulder. You see a sudden change of the waveform here and a sudden decrease of the amplitude over here. This is called conduction block. And conduction block is very simple. If you have all the motor nerves and neuromuscular junctions normally, if you stimulate distally and if you stimulate proximally, you have the same shape, same size compound muscle action potential. If you have, if you lose axons or motor neurons, for example, 2 out of 3, these CMAPs become smaller than normal. In this case, about 1/3 of the normal, but they are equally small. And conduction block is if you stimulate distally, almost normal finding. If you stimulate proximally, the response is very small. This means the motor nerve over here is in continuity. That means there has to be a conduction block on the way to reach to the distal side. So that's the definition of conduction block. And in the disease process, this is mostly caused by demyelination. Demyelination means that inflammation can be treated with some anti-inflammatory or immunomodulating therapy, including IVIG. That's a hint to the development of in my case. So this is before IVIG, significantly atrophied hypothalamus that you can see. This is after IVIG for 3 times, and then hypothalamus becomes almost normal as well as muscle power. So without sensory involvement. And at the same time, these patients do have prominent fasciculations. So this is a sort of a treatable motor neuron disease. And this is another case published in Annals. And these cases do have mostly almost 100 motor conduction block, but using the same stimulation site in the antidromic sensory studies, these sensory conductions are entirely normal. So it is a puzzle still not resolved. So, but this is very peculiar, but it's true. So conclusion here, Awaji criteria are useful for early diagnosis, just confirmed by meta-analysis. What is important is fasciculations and unstable motor units. And electrophysiological findings are very important. The conduction block is a key to the diagnosis of treatable conditions such as multifocal motor neuropathy. And new disease-modifying therapy should employ patients with early stage. So I'd like to just mention some of the examples of these early diagnostic criteria. And yes, I did not mention about growth factors, which Professor Jacobs will talk later on. But for example, disease modifying includes riluzole, and riluzole is very famous and used almost worldwide. But the survival is prolonged, very small, about 90 days. And so you cannot see the clinical changes of the symptoms of the patients with riluzole. And for example, for SOD1 mutation, intrathecal gene therapy is becoming very promising. And we developed ultra-high dose methylcobalamin. This is a derivative of vitamin B12. And we've just finished the pivotal clinical study for approval in Japan. And also Japanese company has developed edaravone as one of the disease, the symptoms ameliorating agent, but they do not have any evidence of disease modifying. So NIPPV and gastrostomy is also important. So edaravone has become marketed worldwide, and this is the study from the Japanese group. And they employed rather early patients. Criteria is 2 years after the onset. And this is ALS-FRSR. This is a symptomatic index of the ALS. And this is edaravone group, and this is control. There's very modest but significant changes in the ALS-FRS. This is this led to the approval of the drug, at least in Japan and the US. And European authority is requiring more longer disease process study. And so many European countries are not officially approved yet. And this is what I mentioned, the gene therapy for SOD1. The final outcome of the clinical study has not yet come, but this is also promising. And I like to mention some of the homocysteine levels in ALS. And the homocysteine levels are rather high in ALS. And nobody knows the answer, but that's been observed. And methylcobalamin is a homocysteine decreasing agent. And this has to be methyl. There are a number of vitamin B12 compounds, including hydroxy or cyano or various others, but methyl has to be the one. And this, if you have a huge dose of methylcobalamin, homocysteine levels will go down. And this is a long time ago. I did a study, so I used low dose group and high dose group, and this is 0.5 microgram per day. This is 25 milligram per day intramuscular injection. And this is a sequential value of compound muscle action potential of the patient. In high dose group, there seems to be a very significant proportion of responders here. So this led to animal study. Again, there are high dose group responding. And so drug company ASI has had begun a very long-term study of the high-dose methylcobalamin in this disease. There are 3 groups: placebo, medium dose, and high dose. And although this trial spent more than 10 years, and they did have some outcome of survival, which is not as easily obtained in these in recent studies. But this is the final result. The total, there was no difference between the survival in the placebo, medium dose, and high dose group. Also ALS-FRS-R. But if you limit those patients who had been diagnosed and entered to the study within 12 months, there has been prolongation of the median prolongation of the survival, more than 600 days, which is much, much longer than Riluzole. And also there's much preservation of ALS-FRS drop in the high dose group. Unfortunately, this is a sub-analysis of the study. The Japanese authority required phase 3 final pivotal study. And this was just accepted as late-breaking abstract for World Congress of Neurology, which WFN organizes, and the Italian Neurology Society organizes. And this is an online meeting, and so you can still join. And this is a kind of world premiere of the result of the study. And this is Japanese government funded and pivotal trial for approval, entry 12 months after the onset, and use of updated Awaji criteria for the first time by the authority. And this is, sorry about Japanese names here in Chinese characters. And but we had more than 20 participating institutions in Japan. This is Japan Early Onset Trial for ALS, the JET ALS. And this is the final result. There's a very significant difference between the high-dose methylcobalamin and placebo group at week 16, as short as 4 months. So this was successful, and the Japanese authorities will probably approve this as soon as possible. And this is changes in the homocysteine concentration. There's none in the control group. This means there's no decrease of homocysteine at all. But for the treated group, there was a big drop of homocysteine as expected. Some other scores also showed some tendency toward in favor of methylcobalamin. So this is the most important message for me. Join us online, World Congress of Neurology. And because this is online, and so you don't have to go over to Italy, but you still enjoy the atmosphere of Rome with a virtual environment created by PCO. And so there are a number of late-breaking abstracts coming on, not only those from ALS. So what I like to mention about ventilation, NIPPV has been most effective measure to prolong life. And so there has been some attempt for diaphragmatic pacing, but the follow-up, some of these negative. And so nutrition is also important, and gastrostomy is also recommended. So conclusion here is Riluzole and Edaravone are approved in Japan, and several promising pharmacological agents, and disease, including disease-modifying therapy, and NIPPV and nutrition are very important. So I'd like to stop at this moment, and acknowledgement goes to my colleagues or my mentor, Jun Kimura, and other people here under the cherry blossoms. Thank you very much for your attention.
Thank you. Thank you, Professor Kaji. We move to second presentation. And for question, in the last webinar, I present Dr. Jacobs. Dr. Jacobs, Professor Jack Jacobs from USA is President and Chief Scientific Officer, Angiogenesis Medicine in Las Vegas, Nevada. Dr. Jacobs received his BA's degree from Davidson College, North Carolina, and a PhD in molecular biology from Washington University. He has been involved in the healthcare industry for over 25 years and has extensive expertise in supervising drug manufacturing operation and the design and implementation of preclinical animal trials and US FDA authorized clinical trials. He started a new department of biological chemistry at the pharmaceutical company, Merck, and for 15 years worked at the Chief Science Officer of Cardiovascular Biotherapeutics, where he oversaw FDA authorized clinical trials in which the angiogenesis drug candidate, human VEGF, was tested in a number of medical indications, including coronary artery disease, diabetic foot ulcer, and peripheral artery disease. He has been continuing these drug development activities with VEGF at Angiogenesis Medicine, concentrating on the potential role of therapeutic angiogenesis to treat neurodegenerative disease, including Parkinson's disease. We move to your presentation. The flowers is yours. You present your presentation. You present.
Okay, thank you.
Amyotrophic lateral sclerosis, potential treatment, utilization, therapeutic angiogenesis. Thank you.
Thank you. I'm going to share my screen.
Yes, is okay. Is shared? Yes. Slideshow.
Slideshow. Okay.
Slideshow, yes.
Okay, 1 second. Okay, is that, see that okay?
Yes, it's okay. Thank you.
Okay. Thank you for the introduction. And Professor Kaji, I really did enjoy your talk and very promising, the JET ALS. I think I will sign up for the October symposium there in Italy. Enjoyed hearing that. And also, I'm a fan of Ohtani, the baseball player too. He is amazing. He's the first player since Babe Ruth to be both a starting pitcher and an amazing home run hitter. So he's been very popular here as well as in Japan, and I enjoy watching him play for the Angels. I'm going to talk about a potentially new approach to a possible treatment for ALS. This is not really on the radar screen yet, but I'm going to try to go through why we think using a drug which stimulates angiogenesis. So this is a growth factor that stimulates the growth of new blood vessels. And I'll show you a little bit of data. First, talk about what angiogenesis is, natural process in our body, how we've used it in humans in the heart, and then move on why we think it may be a potential treatment for ALS. So this is ours and a number of medical researchers, some at the University of Southern California, believe that inadequate blood flow, disruption of blood flow to the brainstem and spinal cord initiates ALS, leading to motor neuron cell death. So initiating event, lack of blood flow to specific areas of the brain. So just to remind you, there are over 75 human diseases that result from a lack of blood flow. Some are obvious: coronary artery disease, stroke, but there's emerging evidence that a number of neurodegenerative diseases, such as ALS, Parkinson's, and Alzheimer's, may be initiated by a lack of blood flow. So let me just mention now this process of angiogenesis. We use it all the time. It's the growth of new blood vessels from existing blood vessels. Here's an example using in wound healing. So let's look at this scraped elbow, look underneath the injured area, let's say the scab on that skateboarder's elbow. So here's angiogenesis healing that wounded area. These are brand new blood vessels stimulated in part by the growth factor that we'll be developing, and I'll talk about that growth factor in a second. But these new blood vessels bring in nourishment to the wounded area and regenerate this area, whether it be skin, sweat glands, hair follicles, nerves. You get complete regeneration of this area. So this is the biological growth factor we're developing. It's called FGF1, fibroblast growth factor 1. It's probably the most potent stimulator of new blood vessel growth in our bodies. And importantly, what's unique about FGF1 is it can stimulate both angiogenesis and small arteriogenesis. So it's a mitogen, not only for endothelial cells, but also for smooth muscle cells. And I was involved in purifying this protein from the brains of cows when I was at Washington University in St. Louis a few years back. It's most abundant in our brains, and it is both an angiogenesis, arteriogenesis, and also is a neurotrophic factor. It just shows that we're not using cow brain FGF1 anymore. This is, we produce FGF1 in an FDA licensed facility. That's human FGF1. This is what we'll be using in the upcoming clinical trial, which I'll talk about at the end of my talk. Okay, this shows how this growth factor, FGF1, initiates angiogenesis. So upon injury, say when that skateboarder scraped his elbow, FGF1 is produced in the body, and it binds to FGF receptors on existing blood vessels, as shown here. So kind of a key going into a lock. This causes endothelial cells to be activated. They start dividing. You get degradation of the basement membrane. And here we have a tube, the beginning of a new blood vessel, and then elongation growing into this injured area to bring blood perfusion into the injured area to begin regeneration, in this case, of skin. Now, one very important point I'm going to make is that upon injury, or in chronic diseases such as heart disease, we know there's an upregulation of the receptors for FGF1. So this causes a directional growth of the blood vessels into the area, the injured area where it's needed. And you don't see an upregulation of receptors on this part of the blood vessel. So you don't get growth of blood vessels in healthy tissue. So I'm going to show you where we've injected FGF1 into the hearts of patients with severe heart disease. You get robust new blood vessel growth. But if you do that to a normal patient or a normal animal, you don't see growth of new blood vessels. So it's only in this injured state do you see about a 10 to 100-fold upregulation of the FGF1 receptor. Okay, before I get into ALS, I just want to show you we have done 4 separate human trials in patients that are called no-option heart patients. That is, they can't have a bypass, they can't have stents put in. So they're basically no-option heart patients. And I'll show you a video where we, through a mini thoracotomy, injected the FGF1 directly into patients with severe heart disease. And I'll show you, or the cardiologist will show you, an angiogram where we can see a dense network of new blood vessels forming after injection of FGF1 into the heart. And rather than me showing you the ABC News, the national news group here in the US, went to one of our clinical trial sites. We had one at the University of Cincinnati. They actually were the highest enrolling, and interviewed the patients. But in this news clip, I want you to pay attention. The Lynn Wagner, the cardiologist who ran this trial for us, points to the new network of blood vessels formed after injection of FGF1 into these patients' heart. So I'm not going to talk through this news clip. It's a minute long, but I'll be back afterwards.
But since receiving an experimental treatment for his blocked arteries, his pain is gone.
I really feel great.
Duke was one of the first heart patients in the country to be treated with a protein actually capable of growing brand new arteries. The genetically engineered protein is injected directly into the heart. Within days, a network of new vessels begins to grow around the blockage, increasing the blood supply. Dr. Lynn Wagner showed us the changes in one patient's heart.
We see a small, narrow main artery and not very many secondary and tertiary arteries. This is after the treatment. What we're now seeing is new blood vessels growing here off the end of this artery.
And the patients themselves?
Symptomatically, they're improved within a couple of weeks of the treatment.
Just ask Constance Donnelly.
Oh, I feel wonderful. I've never felt so good in the last 5 years.
But doctors already see potential in other cases where the blood supply needs a boost, such as strokes and diabetes.
So we're continuing this work in the heart. We'll be initiating phase 2 trials. But let me now turn to some other corroborating evidence, which gives us hope that this growth factor may have potential use in treating ALS patients. And this is animal data where we looked in animals who's given a stroke to and looked at recovery of function and growth of blood vessels in the brains of these animals after intravenous administration of FGF1. And Just want to add that we know that FGF1 can cross the blood-brain barrier. There's a receptor-mediated mechanism for transport, similar to insulin, of FGF1, receptor-mediated transport into the brain. And from animal studies, about 1 to 5% of an IV dose can get into the brain. So we don't have to administer the FGF1 directly into the brain. So in this animal model, it's very simple. We tie off middle cerebral artery and in a mouse brain. We get a stroke, and then we treat these animals for 2 weeks with the FGF1. Decreases the stroke volume. So here's brain slices. We see this is the stroke area, and these are animals. And this is after the animals are given an acute stroke. We let them recover for a few days, and then we treat with FGF1 for 14 days. You can see here the regrowth, the healing of the stroke area. And we can look in this area and look at new vascular growth. So here's normal brain. This is in the microvasculature, capillaries, decimated after we give the animal stroke, and then regrowth with intravenous FGF1 treatment after 14 days. Placebo-treated animals, you can see they're trying to restore their vasculature, but disordered growth is actually hemorrhaging, and the animals aren't doing nearly as well as these animals. We infer we're getting functional neurons by putting these animals, this is a rotating bar, motor skill testing in mice. Normal mice hang on quite well. Here's our FGF1 treated stroke mice. Not quite as well as these guys, but here's the placebo group. They're falling off this rotating bar. So I just wanted to show you this, that in animals, after IV dosing, we can initiate new blood vessel growth and infer functional neuron growth after FGF1 treatment. Okay, so let me go through some of the evidence that this angiogenic growth factor might have potential for ALS treatment. And we have submitted this to regulatory authorities, and we've been given approval to start a phase 1 kind of proof of concept trial, which will be starting in 1 to 2 months. So this was the first indication. This is not our work. This was done by a group in Belgium, Flanders Hospital, published a while ago. This was 20 years ago. First hint that perhaps angiogenesis may have a role in ALS. And what these researchers did, they basically knocked out another angiogenic growth factor. So VEGF, it's vascular endothelial growth factor. Similar to FGF1, FGF1 actually induces the synthesis of VEGF. So FGF1, our growth factor, lies above this growth factor in the angiogenesis cascade. And VEGF also stimulates only the production of capillaries. It's not a smooth muscle monogen, so there's no arteriogenesis with the VEGF. And in fact, VEGF was tested in the heart, very similar to what I showed you with FGF1. And they had to stop the trials because the new vessels formed by VEGF were a bit leaky and caused edema in these heart patients. But again, in this study, they knocked out VEGF. So they knocked out the production of VEGF, specifically in the spinal cord. And what they found, this is a summary of that paper, they had low levels of VEGF, impaired neural perfusion, chronic ischemia, and specifically there was only motor neuron degeneration, no sensory neuron degeneration, and also muscle weakness. So the 2 hallmarks of ALS. And they proposed that both the ischemia as well as insufficient neuroprotection led to the ALS in these animals. Let me just show you another bit of data in that paper. So these are the knockout mice. They knocked out VEGF expression. And basically only about, I think, 40% of the animals lived. Most of them, 60% died at birth. But the 40% who lived, you can see here, after about month 5, they started developing this motor skill abnormality. So here's a very large difference between these VEGF lacking animals and the normal animals. And again, showing a lot of the hallmarks of ALS in terms of specific for motor neuron and also muscle degeneration. So this work was extended. People looked at VEGF expression in patients with ALS. ALS levels were down. Tried treating with VEGF in some of the animal models of ALS, had good results. And then just recently, group published results of the first clinical trial. And here they, since VEGF doesn't transport across the blood-brain barrier very well, they used intracerebral ventricular catheter. So they had a pump, pumping the VEGF into the brain in ALS patients. It was a small study, 18 ALS patients, and it was pretty diverse patient population. Some, I think 4 of the patients had genetic forms of ALS. Some were rapidly progressing. Mainly looked at safety and tolerability and pharmacokinetics. And wasn't powered for efficacy, but they did some efficacy, functional ALS testing. They did vital capacity measurements. They did a European quality index. Main conclusions, they did 3 dosing groups, only the highest dosing group of 2 micrograms per day. And this was carried on for 3 months. And then some patients after the end of the clinical trial actually got the VEGF for, in some cases, a number of years afterwards in an open label extension. But only at the highest dose tested could they detect VEGF in the cerebral spinal fluid by lumbar puncture. Unfortunately, they showed no statistically significant improvement in any of the efficacy testing, which they did. And also, unfortunately, so there were eight patients in this high dosing group. 1 had a problem with the insertion of the catheter, but 3 out of the 8 patients suffered pulmonary embolism. So 2 of those patients had to be removed from the trial, and 1 actually died. And the authors tried to stress that embolisms are common in ALS patients, especially those with leg weakness. And all the patients who suffered a pulmonary embolism in this clinical trial had severe leg weakness. So they thought they couldn't exclude that VEGF was responsible for the pulmonary embolism, but they had what I thought was a decent explanation why this might not be a result of VEGF and why VEGF should still be looked at. Okay, so let me turn now to our growth factor, FGF1, and show you some data in ALS mice. These are transgenic animals. They have a mutated human superoxide dismutase gene put into them, and it's a very classical animal model to test therapeutics before they get into the clinic. And just to show you, this is not our work, this is work from others. We're looking at spinal cord imaging of these ALS mice. They showed a lower density of capillaries between a normal mouse and an ALS mouse. And here is work done by academic research group looking at FGF1. This is same molecule, human FGF1, that we'll be taking to the clinic. Looking at a subcutaneous dosing regimen, looking at time to paralysis in these ALS mice, and a modest improvement in terms of the mice time to paralysis delayed by the subcutaneous FGF1 treatment, 1 microgram per kilogram. Again, sub-Q, and again, about, I'd say 1 to 5% of this dose is getting into the brain. Looking at survival times in the ALS mice, again, a modest improvement in survival with FGF1 against subcutaneously. So placebo versus FGF1 treated animals. Then another study looked at IV-administered FGF1 and looking at motor neuron regeneration, looking at the number of L1 motor neurons in these animals. And this showed a statistically significant increase in these L1 motor neurons. So here's normal animals, ALS mice, and then the mice that received FGF1 intravenously. So with this background of data, we performed some toxicity studies with FGF1. We went to very high doses in both dogs and mice to show that FGF1 was safe being administered intravenously over a 2-month period, and submitted that data to 2 regulatory bodies, the US FDA and also to the regulatory bodies in Mexico. FDA liked our data, but they wanted us to do 1 additional toxicity study, which we're doing now, to show any effects. They were really worried about the retina IV dosing of this angiogenesis. They wanted to make sure there's no neoangiogenesis in the eye, so we're doing that now. But in Mexico, they said you can start treating ALS patients with this drug. Follow such things as tumor markers. We'll be doing retinal photography to make sure there's no neoangiogenesis in the eye. We'll be studying 3 ascending doses of the drug. No placebo group. All patients will receive a 1-hour intravenous infusion of FGF1 5 days a week for 4 weeks. And we're going to administer this through a central PICC line. So this is a line that gets up close to the heart, and we think that will aid in getting the FGF1 into the brain. Mainly safety will be looked at, but we'll also be monitoring effectiveness with the various functional criteria source for ALS. We're going to be looking at levels of FGF in the cerebral spinal fluid, both at baseline and after dosing, and there'll be patients will have follow-up visits for up to a year. We've also just gotten some really recent data, which I think is going to be very important, that in mice anyway, we can deliver FGF1 intranasally and get significant quantities into the brain through what we believe is the olfactory nerve. So upper nasal cavity and then transport into the brain. So this obviously would be a lot easier to administer to patients, and we're now going to be doing a toxicity study to make sure that the FGF1 doesn't damage anything in its transit into the brain. And actually there are devices, commercial devices, which can deliver growth factors into the brain. This was used in a clinical trial looking at insulin delivery into the brain with Alzheimer's disease patients. Now the hospital, people say, Jack, where are you going? You're going to a small clinic in Tijuana. No, we're going to probably one of the finest hospitals in Mexico. It's in Monterrey, Mexico, Hospital Zambrano. New, modern, well-equipped. We have a clinical trial research organization that will run the trial down there to FDA standards. PI Hector Martinez is published in ALS literature. He's trained at the National Institutes of Health. He'll be the principal investigator for our study there. And as usual, we'll pay all the costs. And this trial is going to begin. We've been approved by the Mexican authorities and will begin hopefully, I think in 1 to 2 months. So thank you for your attention. This first trial is going to be a small trial, but if it's successful, we'll expand it greatly to phase 2 trials at multiple sites. You can sign up or follow our progress at our website here, zgm.care. We hope to be getting the US study next year after the successful completion of the animal tox study that we're doing now. You can email me at my email address here. Zicha means it's Ukrainian. One of the founders of our company is Ukrainian. She named it. Zicha means life in Ukrainian. And that's my email at zichamedicine.com. So thank you for your attention, and we'll turn it over now to our moderator for some questions and answers.
Thank you, Jenny. Thank you, Professor Jacobs and Professor Kaji, for your interesting and didactic presentations. We move to a question. We have a questions. Why, especially in diagnosis, why deficit Weakness is frequent in extensor muscle in SLA.
Excuse me, could you repeat that question again? Sorry.
Why weakness,
Yes.
ALS, is more frequent in upper limb, especially in extensor muscle?
That's a very good point. There was a statistical study of the onset part of the body in ALS, and I think upper limb distal muscles are the most frequent site for the onset. But there are a number of variations in the pattern. And of course, extensor seems to be more weakened than flexors in most of the cases. But I really don't know the reason. That means that's a very good question anyway.
Yes, because they use it for mimics inclusion myopathy. We have
yes, IBM, yes.
Yes.
FDP is weaker than FDS. So that's a very good point, but probably nobody has the answer to that.
Yes. Please, Professor Kachi, what about inflammatory markers in cerebrospinal fluid?
Oh, yes.
In ALS.
I think there are a number of disease stages in ALS. For example, the rather slowly progressive early phase and rather rapidly progressive later stage, and at least neuroinflammations takes part in the later half of the process. And so, depending upon the disease process, anti-neuroinflammatory agent, possibly the growth factors that Dr. Jacobs has mentioned might play a role. So I'm not sure about the entire population of ALS do have that significant neuroinflammatory process, but IVIG is so popular in Japan for the treatment of multifocal motor neuropathy. And some patients with a proven ALS would like to have that. And then we've tried that. And some of them do respond to IVIG. So I think that's the later stage of the neuroinflammatory stage. But there has been no solid evidence for the efficacy of IVIG anyway, for ALS. So neuroinflammation is a very important aspect anyway.
Yes, thank you. And how to explain increased iron deposition in corticospinal tract, especially in motor cortex in MRI?
You mean what's the clinical utility of that MRI? Yes, that's something being explored in the past, and so some cases do have para at T2 weighted image of corticospinal tract, especially at the midbrain and some very dense area of the MRI image. But that is not necessarily earlier than the clinical development of motor neuroscience. So some of them might be useful, but most of the cases, the diagnostic process is exclusion and collaborate with EMG. So MRI is sometimes useful, but not always.
Yes, yes, especially to eliminate mimics ALS.
Oh, yes, yes, that's true. That's a good point.
Yes, yes. We move to other questions. Sri Rama, I saw FGF1 used for coronary artery obstruction, and the patient was well after the same. Is FGF1 used in any patient for any neurological condition you mentioned? Question for Professor Jacobs.
Thank you. FGF1 has been looked at in acute stroke models, and there was some issue in the way it was delivered. It caused hypotension, so those studies did not continue. We've also been given approval to start a trial with intravenously administered FGF1 in Parkinson's disease patients. And we actually have more extensive animal data with Parkinson's. We have in toxin-induced Parkinson's in both rodents and, importantly, primates. FGF1 improves motor skills, and also in the primate model, we could show a regeneration of the dopamine-producing neurons in the substantia nigra, and also lowering of the alpha-synuclein levels in those animals. So that, I think, is the strongest data, animal data we have. And again, we've been allowed now to go into humans, and we'll be starting that trial pretty much concurrently with the ALS trial. But it's the same growth factor that we use in the heart. Human FGF1, same protein.
Yes, yes, thank you. Is there any autophagy biomarkers in ALS? For Professor Kaji.
There has been attempt to make one of those biomarkers, but as far as I know, I don't have the exact answer to that. Autophagy is a very important disease process, but not limited to ALS, but also other conditions as well, neurodegenerative.
Yes.
Sorry, I don't have the answer.
Yes, yes, thank you. And actually, we don't have validate biomarkers in ALS.
Yes, that's true. There had been an attempt to intrathecal measurement of TDP-43, but the amount that you measure is very, very small. And there had been a lot of overlapping between the control and the disease groups. So, but those attempts has to be made, because earlier the diagnosis, the better the outcome in clinical trial, probably.
Yes. Other questions? What about use glucoamine in treatment?
Use of what? I don't see the what are the glucosamine. I see. I don't know the answer here. Sorry. Glucosamine had been used as supplement in general, and maybe somebody has to do clinical trial using Awaji criteria, I hope.
Yes. And about what about advanced in therapeutic in SLA, especially familial SLA and in young patients?
SMA had been largely solved by the siRNA, the inhibiting expression of the survival motor neuron and abnormal survival motor neuron gene, and some other genetic treatment had been developed. And so I think similarly, a genetic form of ALS do have very promising agent, possibly SIR, including siRNA, including the one that I mentioned.
Okay, yes. And about edaravone.
Edaravone is true that edaravone is useful in a very small population, because that company had repeated more than 6 times of clinical trials for short runs. And they did enrichment. Enrichment is just narrowing down the enrollment conditions to get the final results of significant difference. I think that might be a way to reduce the time consumed by the clinical trial. But if you do that too much, then there's some problem of interpretation of the result. Edaravone seems to be slightly effective, but there's no information on survival. So that's the status.
Yes. Yes. About administration, intravenous edaravone or intravenous administration?
Yes, intravenous, yes. The company is now developing oral preparation of edaravone, but that had not been released yet.
And what about renal function?
Renal function, yes. Yes, there had been a significant proportion of renal dysfunction after edaravone, so you have to pay attention to that.
Okay, okay, yes. And for Professor Jacobs, what about FG-F1 and physical activity? We have in SLA, a patient who do a lot of activities can have SLA, is a factor environment, physical fitness, athleticism, or player of soccer. There are high levels of patients who have SLA by hyperexcitability of motor neuron. What about FG-F1 and physical activity?
That's a great question. I'm not sure how to answer that, but we know the physical activity stimulates angiogenesis. So we know, especially in Parkinson's disease patients, exercise increases cerebral blood flow, stimulates angiogenesis in the brain. So that's important. I think we're going to look at our drug first in ALS and Parkinson's patients where we have approval and see how it works there. But there are certainly other indications that could be looked at. For example, CTE, the traumatic encephalopathy that soccer players get or boxers get, that is certainly within the realm of being initiated by a vascular function. So we have other indications which we can try, but we can't do everything at once. So we're going to start with those 2 indications first.
Okay, thank you.
Yeah.
Another question. Can colchicine be used as an anti-inflammatory to stop the development of ALS?
I think that's a question to me. I think colchicine has not been tried as far as I know. That might be good, but I don't know the answer. But that has a lot of limitations because of the adverse effects. But I think that's mainly a drug for gout. And maybe drug re-evaluation might be very easy and maybe good. So why don't you try that?
Yes. Professor Kaji, in your practice, what about psychosocial support program for caregivers, SLA, ALS?
Psychosocial management, you mean?
Yes, yes.
Yes, that's very important.
For caregivers, especially, and patients, yes.
Because Japanese don't have, Japanese are not very much religious as you are. and so we don't pray. But there has to be some way of relieving those patients. And so palliative therapy is now being attempted as a medical insurance system for those ALS terminal phase patients. But usually, all occupational groups have to collaborate together for the patients. And for example, I usually put as the eye line level of the eye together with the patients every time I make interviews. And then I spend as much time as I have with the patient. So I think religion is very important in that sense.
Mm-hmm. Yes. And what about COVID-19 and ALS? Yes. You have a patient.
Neuro-COVID project is now going on in WFN, and there are a number of possible consequences of COVID-19 on neurological system. The most famous one is stroke, embolic stroke, and some of the depressive state are now being called as part of brain fog, and some others. And so almost more than 100 years ago, we had a pandemic of so-called Spain flu. That's influenza. And at the time, several years later, after the pandemic, we had encephalitis lethargica, or von Economus encephalitis in young patients. And so we have to watch for that. And so we have to distribute truly effective vaccines all over the world, irrespective of the economic status of the countries. That's my strong point. I think WFN has to work with WHO.
Yeah. And for treatment, especially ventilation tracheostomy, how you convince patient to accept ventilation tracheostomy?
It's up to the patients. We do not recommend that to all the patients. Very rapidly progressive combined with front temporal degeneration is not a very good candidate. Young patients are mostly limited to motor neurons, and they have a good capacity of communicate with personal computers, good candidates for them. So it's up to the patients. But the social insurance system in Japan covers all the expenses of artificial ventilation. So about 1/3 of the patients choose that condition.
Yeah. I think I think we finished the question. Thank you very much,
Thank
Professor
you, Professor
Jacobs
Nisserine.
and Professor Kaji, for this interesting presentation. Thank you, Mr. Shahidi.
Yes, thank you. Thank you, Professor Lohab. Thank you, Professor Kaji and Jacobs. I would like to thank everyone who contributed in any way to the success of this rich and impressive series of online briefing experts in clinical neurology and clinical neurophysiology. IRB also inform you that certificate of attendance will be sent to all participants either throughout Zoom, Facebook, LinkedIn, YouTube, or Nora TV. I also will let you know that we will organize the next Moroccan and African Congress of Clinical Neurophysiology from November 19 to 21. with French and English speaking sessions. More information will be posted soon. Do not forget to register and join us at this Congress of high scientific level. Experts from all over the world will join us. Thank you again for joining us for this successful event, and seeing you soon. Thank you so much.
Thank you.
Thank you.
Bye-bye. Bye-bye.